Lab diagnosis. In lab diagnosis, we will see the microscopic examination, culture, the serology part, molecular part. The samples which are offered for this laboratory diagnosis are CSF, sputum, bronchoalveolar lavage, lymph node aspirations, blood, urine, biopsy samples, and expressed prostatic secretions. So in this microscopic examination, we can do India ink, in which we use an India ink dye, and there we can see under the microscope that budding yeast cell will appear as a starry night appearance. And the second thing we can use is mucicarmine or Asian blue staining. And then there is another stain which is called as Fontana Masson stain. So here we can see it's an India ink preparation, and which are encapsulated budding yeast on an India ink preparation can be seen. So this slide, in the previous slide, we have seen that there is a typical starry night appearance in an India ink preparation. And in this slide, we can see that the histopathological specimens, the stains used are the mucicarmine and the Asian blue staining to demonstrate that the yeast form possesses a polysaccharide capsule. The Fontana Masson stain, it is not specific, but it can demonstrate the production of melanin. So this is a picture of an Gomori methamine silver stain, and see we can see an budding yeast cell with a typical budding of a daughter cell. Represents a simple budding. Now the culture. The CSF should be collected in large amounts, 20 ml, and centrifuged. And two separate dextrosaccharides, one each at 37 degrees centigrade and the other at 25 degrees centigrade, should be inoculated and kept in the BOD. That is the biological oxygen demand incubators. So we can inoculate it in a sapro's dextrose agar, brain half infusion agar, the bird seed agar, or capic acid containing media. Colonies can appear in 2 to 5 days, and they are creamy white, shiny in color. The colonies are black on tiger seed agar, and they appear in 5 days. Confirmation can be done of the cryptococcus by a UES test, which is positive inositol assimilation, and negative nitrate assimilation test. So in this slide, you can see the white mucoid colonies due to the polysaccharide capsule in the routine laboratory aroused. Within 48 hours, we can see the first one is the saprodes. So this is an STA basically, that is called as the saprose dextrose. And in this, we can see that there is a typical white mucoid colony. There we can see that there is a production of melanin in a niger seed agar, or a bird seed agar, or a stab agar. So stab agar, this is a picture of a stab agar in which there is a brown, there is an appearance of the brown colonies due to the production of the melanin. There is a biochemical reaction which is representing the nucleus as positive, and it is showing pink in pinkish red in color. So rapid nucleus test for the Cryptococcosis neoformans is positive. So in serology part, we can detect the antigen or the antibody in the cryptococcosis. So first of all, the antigen, it can be in the form of capsule polysaccharide or the protein antigen. The method being used for the polysaccharide antigen detection are the latex agglutination test and the ELISA, whereas for the protein antigens, it is ELISA and the Western blot. The various antigen detection kits, that is the latex agglutination kits, available in the market are there's an Cryptola, Microimmune, Immune, Calas, then Equilitet, then Pasteurix Cryptococcus, and Murex Cryptococcus. And the samples which are being used here are the CSF and the CSF serum and the BAL, that is the bronchoalveolar lavage. So these are the different kit names which are being introduced by different companies, and these are being used for the antigen detection in cryptococcosis. The false positive results can be obtained in the antigen detection in cryptococcosis by the presence of a pneumatide factor, trichosporin deadly infection, somatococcus mucilaginous, capnocytophaga, candimorsus, and capsula pneumoniae. These can give the false positive results if the patient is infected with any of these diseases. And because of the presence of the antigens, um, antigens in the body, these can give the false positive results. So the false negative results can be due to low concentration of the antigen, the protein phenomena, and poorly capsulated strains. Technical error can be there. So that means so these are the false, some of the false positive and false negative results which are associated and which we have to be kept in mind before having the final result. So antibody detection in cryptococcosis. And what happens here is sometimes the immunologically paralyzed due to the immunologically paralyzed status of the patients infected with HIV and those who are severely immunosuppressed, the T-cell mediated immunity is specifically not affected. It is specifically affected, and that is why there is a less of the antibody response in our body. So, which is mediated by the type 1 T helper cells and the type 2 T helper cells. So, cryptococcal antibodies is not useful in diagnosing and deciding the treatment because of its poor sensitivity and specificity performance, and it can be positive in the absence of an overt disease also. Last but not the least, molecular typing. It can be done by multi-locus enzyme electrophoresis, DNA fingerprinting, random amplification of polymorphic DNA, then PCR fingerprinting, amplified fragment length polymorphism, then multi-locus sequence typing, multi-locus microsatellite typing, and multi-top analysis. So basically, the children, you can go through this anti-fungal treatment, which is recommended for the cryptococcal meningitis and HIV patients. So there can be induction therapy, consolidation therapy, and maintenance therapy. So in induction therapy, amphotericin B is along with the flucytosine is recommended, and in alternative to for induction therapy is can be amphotericin B plus fluconazole or fluconazole plus flucytosine or fluconazole alone or ipraconazole. And in consolidation therapy, fluconazole is recommended 400 mg per day, and in maintenance therapy also fluconazole is recommended 200 mg per day. So the antifungal treatment recommendation for cryptococcal meningitis in non-human immunodeficiency virus infected and non-transplant patients. The induction therapy includes amphotericin B and plus flucytosine, or it can be amphotericin B only. And in consolidation and maintenance therapy, fluconazole is given, and the dose is decided. So the doses in the previous one is decided according to the regimen, and in antifungal treatment recommendations for non-meningeal cryptococcus, like for example pulmonary cryptococcus and non-pulmonary cryptococcus, the fluconazole is the recommended drug of choice. So the major complications which can occur is the CNS inflammation with increased intracranial pressure, in which we can give corticosteroids with high dose, that is the high dose dexamethasone, and a severe CNS in other CNS sign where sign and symptoms are seen. Antifungals can also be appropriate. Antifungals can also be added. So, the gist of the treatment is that the fluconazole is recommended for mild to moderate infections, and it is the drug of choice for the non-meningococcal cryptococcus. Whereas amphotericin B plus flucytosine combination plus fluconazole can be given in the severe infections in meningeal cryptococcus. So Prognosis, the most important prognostic factor is the nature of their underlying illness, for example, malignancies or AIDS, which contributes to the immunosuppression. Mortality rates in the HIV-infected patients in the developed countries is approximately 10 to 25%, whereas in resource-limited countries, due to the delay or inadequate treatment, it can be increased up to 37 to 43%. So thank you. Here we come to an end for the streptococcal meningitis, and we can clearly see that we have discussed first of all what is cryptococcal meningitis. So what is its method of reproduction, its epidemiology, then its virulence factors, the pathogenesis part, and what are the clinical presentation of a patient, what is the treatment which should be in the induction maintenance phase, and then we have done the prognosis for this disease. Thank you. I hope that you have that you have gathered some information through my this small effort. Thank you.